Hi, I'm going to be explaining the cell transport and dialysis lab. Unfortunately, we can't do it together in class, so I'm going to be making a virtual lab for you to follow. So if you read the introduction, it kind of goes over what you learned in the Amoeba Sisters video, that our cell membrane is a double lipid layer, and it lets certain things in and certain things it doesn't allow in and out, and that's called selectively permeable. And this moving of things in and out is called cell transport. So sometimes things can move in and out without using extra energy, and that would be diffusion, facilitated diffusion, or osmosis. When there's a lot on one side of the cell membrane, then naturally the molecules will diffuse across the membrane from high concentration to low concentration. So that's going with the flow, high to low. Sometimes it needs a little help if it's too big, but all of these don't require energy. If we're talking about water moving from high to low, we call that special type of diffusion, we call that osmosis. And then sometimes you do require energy when you're going from low to high, and so that's called active transport. Now the kidneys are really important. They're kind of on the back side of your body, so it looks like it's in the front, but they're toward the back. They're behind your front organs, your intestines and things like that, and they're in the back. And they're important for filtering waste. So if you look, your blood comes through these kidneys, and then the waste gets filtered out through these ureters, and they go to your bladder, where your bladder holds it until it fills up, and then you pee out your waste. So this kidney, here's a close-up, has all these tubes which basically filter out any waste products from your blood, and then your blood goes back to the rest of your body nice and clean. So if you have kidney failure and your kidneys aren't able to do their job, then you will need to go for dialysis, which is where this machine does the filtering for you. So they basically take your blood out, put it through the machine, it filters out the waste, and then puts the cleaned blood back into your body. And this process takes several hours, I think 3 or 4 hours, and then you have to go 3 times a week. So it's a huge treatment or inconvenience if your kidneys are not working, or you could get a kidney transplant if you are lucky. This is a close-up of what's going on inside the machine. Here's your blood coming through, and then there is something called dialysate, which is flowing on the other side of your blood, and there's a membrane between it where the waste will move from high concentration out of your blood into a low concentration of the dialysate. So it's actually using the property of diffusion to get rid of the waste out of your blood. And so we're going to be looking at a lab that is going to see, it's going to model the dialysis process. So first I have some fake blood, and we'll be testing the fake blood for glucose, protein, and salt. And then we'll be putting it in a dialysate to see what filters out of our blood, and we'll be testing it in 24 hours to see the results. So I'd like you to write your answers on our answer sheet. This first question is asking what is dialysate, and that's right here. if you're not sure, it's written right here. So here it talks about what dialysate is, and it's going to pull the waste from your blood because it has a low concentration of waste, so the waste is going to go from high to low. Okay, and and then we're gonna test our simulated blood for this, and then we're gonna put it in dialysis tubing, and then we're going to put the tubing into a cup of dialysate. So let's go ahead and do that first, and then we will come back to our lab. Okay, so here's my fake blood. Okay, I tried. And then here is dialysis tubing, which is going to be where we put our fake blood. So it's just like a little plastic bag type of thing, but it has a membrane that lets certain things in and not others. So what I'm going to do is I'm going to put blood into this little baggie and then tie it. So I'm going to do that over the sink because I don't want to get this on my computer, and I'll be right back. Okay, so this is my my filled tube of fake blood, and before we move on, we do need to test the blood to see if it has any protein, glucose, or salt strips. So let's do glucose first because that was first on the data sheet. Okay, so let's see what our results are supposed to look like. So if we look back on our lab, if there's glucose, then the test strip will turn pink. Okay. If there's proteins, then the test strip will turn a dark blue to dark purple. And if there's salts, then it will turn dark purple. Okay, so let's go ahead and check that. All right, so here we go back. All right, so there's our glucose test strip. It is pretty pink. Here is protein. Okay, let's give it a second. Okay, so protein turns dark blue to dark purple in the presence of protein. It's kind of hard to see on the camera because I think the camera is picking up some weird tones. And the salt is supposed to turn light to dark purple. Okay, so I know on the camera it's looking like it's more orangey yellow, but actually it is getting darker. So let's go ahead and do this part together. So here are glucose test results. So hopefully you, our test results were that it is, is that it is positive for, okay, I'm going to put just positive, positive, and positive. So it has all three, glucose, protein, and salt. You don't have to have yours red with yellow highlight, but that might help you see it better. Okay. I don't know if it, and then let's go ahead and fill in this before dialysis. So there is urine in the blood, it's telling us. There is red in the blood. By just looking at it, it's red, so we see blood cells. There's protein, so you would put yes. There's glucose, yes, and salts, yes. Okay, and then our initial setup is going to be this is our dialysate, okay, and we're going to put our blood, which is in the bag, in there, okay. So you would draw a picture. Let's see if we can. Okay. So you would draw a picture of this cup with the blood in here, and then you would label where everything is. So we know there's red blood cells inside. We know there's urea inside. That's the waste that's going to come out in our urine. We know there's protein inside, there's glucose inside, and there's salt inside. Okay, this dialysate doesn't have anything in it. It's just water. Okay, so you're going to do your initial setup picture right here, and you're going to just label everything that's inside the blood, and then we're going to go ahead and let it sit overnight and see which of these substances diffuse out of the blood into the dialysate. Okay, and before we come back, go ahead and write a hypothesis. What do you think will move from the blood to the dialysate when left overnight? Do you think the urine will come out of the blood? Do you think red blood cells will come out, protein, glucose, or salt? Okay, so think about what needs to move out of your blood into the dialysate to be removed as waste. Okay, so some of these might be waste, but some of them might not be waste. So go ahead and write that, and then I will come back in 24 hours to show the results. Okay, it's been 24 hours, and here is our blood in our baggie. It's not a blood cell, but it's blood in a baggie. And here is our dialysate, and you can see it used to be clear, but now the dialysate is yellow. And let's take a look at our lab. Okay, so it's you can go ahead and write the color of the dialysis the color before and after dialysis of the dialysate in the blood, and then now we're going to test for glucose, protein, and salt. So let's go ahead and do that. I'm going to go ahead and see. Remember, we had glucose, protein, and salt inside the blood, and let's see if any of those substances diffused into the dialysate overnight as it sat overnight. So I'm going to go ahead and test. So here's our glucose, and I'm going to dip it, and oops, I did the wrong end. Dip it. and I'm going to go ahead and put the test strip. It needs to sit for a couple moments. I'm going to put it here. And then our salts, dip it, and proteins. So I'll go ahead and move this out of the way, and then we'll take a look at our results. So I might have to doctor up these results a little bit, but the glucose, it did turn pink. It did turn pink, and if you remember, pink is a positive, so it will turn pink or red. So what we can do here, and actually here we're supposed to put, originally it turned pink, the glucose turned pink, and then here as well, the glucose was positive, positive, and it turned pink. Okay, now for the protein, it will turn dark blue to dark purple. The protein, it is just wet. I know it looks dark. I know it looks dark purple, but this is just wet, and actually this is not supposed to be positive. So I'm going to tell you the answer, that in the dialysate at the end, this one is supposed to be negative. So we'll say no color change. It's hard to really say because the strip is purple, so I'm not really sure why. I mean, I guess it's supposed to turn dark purple, but when purple turns wet, it does look dark purple, so that is a little confusing. So I'm going to go ahead and let you know that the protein did not move into the dialysate. The protein stayed in the bloodstream. Okay, so, well, not bloodstream, but in the blood bag. Last is going to be, so here it did turn in the blood, it was dark purple. Okay, but in the dialysis result, there was no dark purple. Okay, so last but not least, let's look at the salt. So here's the salt. It did turn a purple color. So let's see what that means. So the salt yesterday, if it turns a light to dark purple, that means it is positive for salt. So in yesterday's blood sample, it did turn purple. Okay, so that's positive. And also in the dialysate today, positive, it was positive, it turned purple. Okay, so it looks like the salt moved out of the bag and the glucose moved out of the bag, but the protein stayed in there. Okay, and you can go ahead and look at the color. You can see if was the urine present in the dialysate. Okay, so let's see, is there a yellow color in the dialysate? Well, I would definitely say there is yellow in there. Okay, is there yellow in the blood? Probably still some. Okay, but that one is hard to say. But definitely the urine moved out of the blood into the dialysate. Is there blood In the dialysate, well, I don't see any blood in here. It looks like the blood just still stayed there and did not cross through into the dialysate. Okay, and then as for the others, you can go ahead and answer these questions if they are present in the dialysate after dialysis. Now, as far as testing the blood, I don't know that we needed to test the blood afterwards. Oh, I see. Okay, so if the protein, for example, if the protein did not move into the dialysate, then it is still in the blood. Okay, and the glucose we know moved into the dialysate, and the salts moved into the dialysate. I'm not sure if all of them moved into the dialysate, so here you can put yes or no, that's okay. But we definitely know that the glucose moved into the dialysate, the salts moved into the dialysate, but the proteins did not. The proteins stayed in the blood. Okay, so go ahead and answer the yes, no on this data table. Fill in the colors of before and after. I was just talking about this data table, sorry. So in this part right here, we know that the proteins were present in the dialysate after. I'm sorry, we know that the proteins did not move into the dialysate. Okay, so right here. So the proteins did not move into the dialysate, but the glucose and the salts did. They did move into the dialysate. But the proteins did not. The proteins stayed in the blood. So you're going to go ahead and fill in the rest of this data table, answer these questions if you haven't already, and then go ahead and answer the analysis questions. Okay, as far as your picture, this picture is going to be the same as the picture that you had above here for your initial setup, okay, which we talked about right here, where there's nothing in the dialysis, but all the substances are going to be in the blood. So the blood, water was in the dialysate, urine, red blood cells, protein, salts, and glucose, those are all in the blood. Okay, and then your after picture is going to show what moved into the dialysate. Okay. Even though like glucose and salt moved, there's still going to be glucose and salt in the blood. Okay, because it doesn't all move out, it just moves until it's even. Okay, and then did the urine diffuse? How do you know? Did the red blood cells diffuse? How do you know? Okay, so if you have any other questions, go ahead and email me or text me on Remind, and I hope you enjoy finishing this lab.